But with that, I'll turn it over to Diana, and thanks everyone for joining us.
Thank you, Gary, and welcome everyone. Thank you so much for joining us today. And thank you so much to our LifeVest sponsors, Blueprint Medicines, Decipher, Genentech, Novartis, and Pfizer for making this weekend possible for our community. And I also want to thank Norman Scherzer, our executive director, for just being who he is. And he's not here with us today, but Laura is representing him. So thank you. I also wanted to make mention real quickly alongside with that, what Gary mentioned, our cooking demonstration. That will be led by chefs Rosemary and Drew Gabe, who will prepare LRG member Rob Taylor's Happy Shrimp recipe. The video will premiere on YouTube at 4 p.m. Eastern Daylight Savings Time today. And so we're so much looking forward to that. And don't forget our Just Do It walk that's happening tomorrow. Don't forget to check out our store to order masks, our Just Cookbook, our t-shirt, and Just Art that has been created by LRG member Manuela Quick. It's amazing art, so please go over to our website to see those things. So without further ado, I have the honor of introducing our esteemed presenters, Dr. Michael Heinrich of OHSU Knight Cancer Institute, and Dr. Jason Slick of the Moores Cancer Center at the University of California, San Diego Health Systems. After the presentations, we will accept questions from the audience, which I will moderate. You can ask questions by using the Q&A feature at the bottom of your screen. Just click on the icon, type in your questions, which the doctors will answer during the last portion of the panel. For those of you who have already submitted questions to me, I will ask some of them of the doctors during this Q&A, and some of the other questions that are relevant to the Oncology and Surgical GIST Specialist panel at 2 p.m. Eastern Daylight Savings Time today. So allow me to turn it over to Dr. Sicklick. Thank you.
Thank you very much. It's a pleasure to be here today, and thank you for the opportunity to present some of our work on GIST. I'm going to make the assumption that everybody attended the GIST 101 session yesterday, and so I'll jump right into some of the research that we've been doing and the potential for thinking about new therapies for GIST. The title of my talk is Targeting Paracrine Fibroblast Support in Gastrointestinal Stromal Tumor. And these are my disclosures. So as you probably know, GIST is targeted by many drugs or tyrosine kinase inhibitors, also known as TKIs. But what we also know is that these fail to cure GIST. The only potential cures for GIST are either surgical resection or surgical resection in combination with drug therapy. But drug therapies alone rarely cause complete responses to therapy. And if you look here at the data from several of the clinical trials looking at imatinib, sunitinib, and regorafenib, the response rates, or the complete response rate, meaning that 100% of the tumor disappeared, and the partial response rate, suggesting that there was at least a 30% shrinkage in the tumor size, show that these are still not where we'd like them to be, meaning 100%. You can see here for imatinib, it's between 50 to 60%. Apologies. And for sunitinib, it's about 7%, and regorafenib about 5%. Having a little trouble advancing slides, so bear with me here. So what we know about many types of cancers is that the milieu of a cancer is more than just the tumor cells. So the cancer cells here are represented in this image in green. But as you can see here, there are lots of other types of cells present within a cancer. There's immune cells, there are fibroblast cells, as well as potential stem cells. And all of these together interact in a microenvironment that supports the survival of each cancer. The focus of my talk today is going to be on the cancer-associated fibroblasts, which are these as highlighted down at the bottom there. So we hypothesized that gastrointestinal stromal tumor cancer-associated fibroblasts may play an unappreciated role in the tumor biology of GIST and may serve as a novel therapeutic target for GIST. From what we know in other tumor types, these cancer-associated fibroblasts have been associated with survival of cancer cells, with promoting invasion and metastasis of tumors, with resulting chemoresistance, meaning that the drugs don't work, with affecting the metabolism of the tumors, as well as the immunity within the tumors. So most of the work that I'm going to present today was done by Danny Yoon from my laboratory. Danny's a PhD from Korea that's been with us for about a year and a half now. So we first thought, if we looked at the stroma, meaning the non-cancer cells within tumors, and specifically within GIST, you can see that there's what I would consider an unappreciated area of tumors, meaning the stroma here, this sort of wispy looking area. And if we look at these cells within tumors, and these are both a KIT mutant tumor as well as a PDGFR alpha mutant tumor, what we see is the red tumor cells marked by KIT expression, but we also see these green cells. Protein marks what are called the cancer-associated fibroblasts, or these fibroblasts that are not themselves cancer cells, but are cells that live within the tumors. So we started off by just asking a simple question of, can we isolate these cells and can we characterize these cells? And we can separate these cells from actual tumor cells. And so here's just an example from where we've separated the cells from a tumor, that PDGFR alpha D842V mutant tumor. You can see here's the tumor cells here, and then there's these large sort of stellate-like or star-like cells are the fibroblasts. And if we look for the markers of GIST as compared to the markers of these fibroblasts, what we can see here is that in our cell lines, the GIST T1 cell line, as well as in that one specific patient case where we've separated here, the tumor cells are stained by KIT protein, with the blue DAPI marking the nucleus of the cell. In contrast, the cancer-associated fibroblasts, or CAFs, have no expression of KIT, so there's no red staining, but they express this green stain, FSP1. Likewise, we don't see that FSP1 staining in the GIST tumor cells themselves, suggesting that this is clearly a different population of cells within the tumor. So we first started out by just asking a simple question, is whether these cells can promote growth, migration, and invasion of tumors. Growth obviously correlates with the growth potential and mitotic index of cells. Migration and invasion assays, where we're doing this in Petri dishes, are essentially surrogates for whether the cells have the potential to invade or metastasize. So if we start off just looking here on the left side of the figures, you can see here that when we grow cells alone, we certainly see growth of tumor cells, and these are marked by green. But when we mixed fibroblasts in with them, we see a significantly larger size of these spheres that form, and this is represented not only in size, but also in number of spheres, as you can see here in the graph. Similarly, if we put the cells into a matrix that is essentially a three-dimensional matrix, you can see here that each dot is a colony of cells, that the number of colonies is significantly higher with the fibroblasts being mixed in with the tumor cells, suggesting that the fibroblasts are aiding the tumor cells in some way to grow. So they're creating a growth advantage. Furthermore, if we can grow the fibroblasts and the tumor cells separately, so we have these inserts that we can grow essentially in 2 different layers side by side from each other, asking the question of, do the tumor cells have the ability to sort of grow through this membrane here towards these fibroblasts that they're trying to have some magnetic pull, if you will, or some type of signals telling them to come towards them. And what you can see here is that when we have tumor cells alone, we can look for migration through or invasion through this in a matrix. And you can see here, this is significantly greater when we have the fibroblasts mixed with the tumor cells, with the tumor cells up here and fibroblasts down here, as opposed to no cells down here. Simultaneously, we can also look at so-called wound healing or scratch assay, where we basically scratch the surface of the cells and look for ingrowth back of the cells. And you can see here again, tumor cells alone, tumor cells after 24 hours, tumor cells with fibroblasts, and tumor cells after 24 hours. And you can see the significantly smaller gap between the cells as the cells try to grow back towards one another. So if we take this to the mice, what we can see here is if we inject mice with tumor cells versus tumor cells plus the fibroblasts in 2 different cell lines. This is a KIT exon 11 mutant cell line, and this is a KIT exon 13 mutant cell line. What you can see here is that the sizes of the tumors are much bigger in the tumors that have the fibroblasts than those that do not. And this is represented by the tumor volumes here. And likewise, this also results in more rapid growth of the tumor. So if you look on the right side, this is growth, this is tumor volume growth over time. And what you see here in the orange is the tumor cells alone, and in the blue is the tumor cells growing with the fibroblasts. Let's look at the expression of these cells as compared to our cell line, our GIST cell lines. And we did what's called a principal component analysis, which is basically a sophisticated analysis to demonstrate that the tumor cell lines are very different than the fibroblast cell lines. And interestingly, one of the most overexpressed markers in the fibroblasts was a protein called PDGF-C, or platelet-derived growth factor C. Why that becomes potentially important is because, as many of you know, or you learned yesterday from GIST 101, after KIT mutations, the second most common mutation in GIST is PDGFR alpha, which is basically the receptor for the PDGF-C ligand. So in the absence of a mutation in this gene, which when this is mutated, it works on its own without having to see this ligand. But in the absence of that, what happens is that one of the PDGF-C ligands, whether it's A, B, C, or D, binds to 2 different receptors. One is PDGFRA, and then the second one is PDGFRB. Well, the focus of the driver of most GIST is this PDGFR alpha. And what we can see here is that really in these GIST cell lines, we do not see expression of the 2 ligands, PDGFRA and PDGFRB, but a majority of this is through the ligand PDGF-C. And it turns out that PDGF-C can bind to the PDGFR alpha receptor. It can also bind to a combination of the PDGFR alpha receptor with the beta receptor. And then finally, while we all think that when the PDGFR alpha is mutated, that all those combine together and it's only mutants with mutants, you can also see combinations of the normal PDGFR alpha with a mutant PDGFR alpha combining together, which can also be activated potentially by the ligand. So with that being said, we then ask the question did this PDGF-C, is this present in the human GISTs? And so we looked at human tumors, and this is a human KIT exon 11 mutant GIST, and this blue again marks the nuclei of cells. And what you can see here is there's a population of cells that is expressing this PDGF-C. Simultaneously, this exact same population is also expressing that fibroblast marker FSP1. And when you overlay the images from all three of these together to merge them together, you can see that the comment that there's a change in color from the red plus green becomes a yellow, suggesting that these are the same cells that are expressing this marker, or they're being expressed at the exact same location. And what this together would suggest is that PDGF-C may play an unappreciated role in the biology of fibroblasts and in GIST. So the next question we asked is, we looked at our GIST cell lines as well as our fibroblasts, and asked the question, is this PDGF-C not only being made at the RNA level based upon that RNA analysis I showed you earlier, but also is it being secreted by the cells, or being basically extruded from the cells from the fibroblasts? And the answer is yes. This is looking at the expression of the protein expression and showing that it's significantly higher in the fibroblasts as opposed to the tumor cells. Furthermore, what we can show is that if we give the tumor cells this ligand, that we can activate this PDGFR alpha receptor here by turning on the phosphorylation or activating this protein. So, which taken together would suggest that this potentially has a functional role. So then we return back to the concept that I showed you earlier, saying that calves promote GIST growth, migration, and invasion. So now we've specifically asked the question, is this through PDGF-C? And so we started off by asking a simple question: if we give the GIST cells increasing doses of recombinant or basically engineered PDGF-C that can be purchased from scientific companies, can we increase the growth of these cells? And the answer is yes. By giving the cells more PDGF-C, that itself can increase the growth of the cell line. If we give PDGF-C to the cells, can we increase the migration and invasion? And you can see cells, and answer again is yes. And this is the graphical representation of the data on the right side here, showing significant increase in the amount of migration and invasion. So does this work in the mouse? So the next step we did was we created a cell line, or two cell lines in fact, that lack or have low expression of PDGF-C. And so these are represented by the green and the blue cell lines. And what we did was we then injected mice with either T1 plus calves, and these are fluorescently labeled tumors, or T1 that lack the PDGF-C in two different models. And what you can see here on the right, this is looking at the fluorescence, and then on the far right is actually looking at tumor weight, is that over time we can follow the fluorescence of these tumors. And what we see is that when we just grow tumor cells alone, we see an increasing amount of fluorescence, meaning that the tumors are growing over time. And we see that if we give them a combination of the tumor cells plus growing them with the fibroblasts, we see a much more significant increase in the amount of fluorescence in the cell in the mice over time. Now, if we abolish that expression of PDGF-C, you can see here in both of those cell lines mixed with the T1s that we see a dramatic reduction in the amount of growth of the tumors. And this is represented here by the actual weights of the tumors. You can see here, these are smaller tumors with T1s. These are fairly large tumors with the T1 plus calves, and that this growth advantage is abolished or diminished with the fibroblasts that do not have PDGF-C.
Jason, I'm sorry, the slide isn't advancing for everyone.
Did I advance?
It's showing for me, but on the live, it's not advancing for people.
Do you want me to stop sharing for a second here?
Yeah.
You can try sharing again. Is it advancing now?
Let's just wait a second and see what people say.
David just said it's.
Yeah.
Yep.
Yeah, it's working now.
Is it working now? Okay.
Yep.
Okay. So I just showed you the data that the PDGFC itself is playing a role in tumor growth. Can you see this? I'll assume yes. So the next question was, does the PDGFC itself play a role in the migration and invasion? And again, if we grow the cells as either as the just T1 cells without fibroblasts, with the regular calves, or with the calves that do not have PDGFC, you can see this dramatic reduction in the amount of migration and an invasion. And this is represented here in the bottom slide again. So migration and invasion, as I mentioned before, is a marker, if you will, for metastasis. And so we next sought to look back in the mice. And so we performed a metastasis assay by essentially injecting tumor cells or tumor cells with fibroblasts into the spleen of mice. And what happens is the blood vessel that drains the spleen, which is called the splenic vein, will then drain into the liver. And this is a mechanism by which you can actually seed the liver with tumor cells. And what you can see here, this is with looking at the tumor cells plus fibroblasts, and then tumor cells with our knockdown of the fibroblasts. You can see here both by just grossly viewing these white nodules or tumors, or by looking at the fluorescence of these tumors, that we can see a dramatic increase in the amount of tumor number and fluorescence when we treat with fibroblasts, and that this is diminished in the absence of PDGFC. To the right here is just a numerical calculations representing showing increasing amount of tumor metastasis and then decreasing with loss of PDGFC. So we know that metastasis is a very complicated process by which cells leave their location and begin invading through the layers of the tumor and able to travel to a distant site. And one of the processes that's thought to be involved with this is a so-called epithelial mesenchymal transition, where cells basically lose some of their tight junctions or their ability to adhere to one another, and they can leave. And this process is often regulated through different types of growth factors. And ultimately, these growth factors, including PDGFC, can lead to downstream activation and expression of different genes associated with epithelial mesenchymal transition. So we looked at several markers of epithelial mesenchymal transition in our models. And we first started off by looking at expression of one of these markers called slug with RNA-seq from human tumors using data that was obtained from Dr. Ron DiMatteo. And what you can see here is that expression of slug is present here in the KIT mutant GIS. It's dramatically higher in the PDGFR alpha mutant GIS, and that it's very low in this, albeit in a small number, of KIT PDGF wild type GIS. Next, in collaboration with Dr. Heinrich, we looked at his PDGFR alpha mutant GIS cells, and we see that if we overexpress PDGFR alpha in these cells, that we see an increase in slug expression. Does this occur through activation with the ligand PDGFC? And the answer is yes. So if we increase the amount of PDGFC in the cells, we see an increase in the amount of slug expression, as well as N-cadherin, another marker of EMT. And what you can see here on the right is that if we knock down expression of PDGFC in our fibroblasts, that we can abolish that increase in the amount of slug expression. So this would suggest that slug in some way is being driven by PDGFC. And without going through all the stepwise details and the additional data proving this, basically it turns out that slug is being driven by PDGFC based upon additional experiments. So the next question we asked was whether slug itself was important in the cells. And so we sort of took the same approach that we had done with the PDGF-C in the fibroblasts, but instead we've now turned to the tumor cells that express slug, and we knocked down or diminished the amount of expression of slug in the tumor cells themselves. And what you can see here is that migration as well as invasion are dramatically decreased by loss of slug expression. And this is the numerical quantification of this on the bottom. Similarly, if we treat this, if we look at the tumor cells alone or the tumor cells with calves, what you can see here is that tumor cells, normal tumor cells with calves, have a tremendous amount of metastasis, and our metastasis model is shown here. If we take tumor cells and we add calves to them, but we've lost that slug expression, this amount of metastasis is diminished. And you can see here, this occurs for both versions of our slug loss of expression experiments. And over on the right here is the quantification of this, again, showing that we can dramatically abolish the amount of expression of slug and ultimately abolish the ability to metastasize. So what this model would suggest is that there is a crosstalk between cells not only that normally would be the cancer cells, but between these non-cancerous cells within the tumor themselves that are secreting this PDGF-C ligand, which is connecting with this PDGFR alpha receptor, which is then driving the PI3 kinase mTOR pathway, which we don't have time to talk about in detail in the tumor cells, which is ultimately leading to activation of slug expression, which then correlates with proliferation, migration, invasion, and ultimately the metastatic potential of these cells. So in the last few minutes of the presentation, we next asked the question is, can we target these fibroblasts? And it turns out that these fibroblasts are fairly insensitive to killing by the standard drugs that we all think of for treating GIST, including imatinib, sunitinib, and regorafenib. You can see here dramatic cell death of the T1 cells, represented in blue here. But when we treat the fibroblasts with these same drugs, we shift these curves over, suggesting that they are less sensitive to treatment with these agents. So we next sought to look for a method to target these cells. And in looking at our RNA analysis of the calves, one of the pathways that, as I sort of alluded to before, that was important also in the tumor cells was the PI3 kinase pathway, was the second most commonly overexpressed pathway in these fibroblasts. And so the PI3 pathway is driven by ligands, including PDGFs themselves, which then this can actually activate the growth of these tumors cells as well as the fibroblasts. So we now took and generated new tumors. This is using a combination of tumor cells, the T1 cells, plus fibroblasts. And we then treated the tumors with either imatinib, gedatolisib, which is a PI3 pathway inhibitor that we identified after doing a drug screen of about 40 different drugs, looking for the most effective one at killing the fibroblasts, or the combination of both. And I don't have time to show you all the culture experiments where we've shown that these are synergistic, but suffice it to say that in the mouse, it also worked as well. What you can see here is that if they're just allowed to grow on their own, this is tumor cells with just control. This is tumor cells with either single agent imatinib or gedatolisib. And this is the combination of both of these drugs treating the tumors. And you can see here, here's the actual tumors themselves, the vehicle, imatinib, gedatolisib, and then the combination of the two. And this is tumor weights here. So what this would suggest is that the calves represent a new therapeutic target in GIST, whereby we can target the fibroblasts with drugs such as gedatolisib, which will then diminish the amount of PDGF-C being expressed. And simultaneously, gedatolisib may also have a role in targeting the tumor cells as well. And this is synergistic with targeting imatinib, which can inhibit KIT in order to therefore decrease this cascade of events that's driving the biology of these cells. So taken together, what this data would suggest is that it's not just about the tumor cells themselves, as we've frequently focused our attention on, is that there are other cells in the microenvironment of tumors that can also drive the growth of GIST. And with that, I'm going to say thank you to all of our funding agencies, including the NIH, the FDA, LifeRaft, Lighting the Path, SDHRA, Phase 1, and acknowledge most of this work, as I mentioned, was done by Danny Yoon, shown here, as well as in collaboration with several other members of my lab, including Amy Tang, Sudeep Banerjee, and Myra Yerba, as well as with Mike Heinrich and Lily Klug from OHSU, and Ron DiMatteo from Penn. And I'll stop there, and thank you very much for the opportunity to present some of our work today.
Thank you, Dr. Sickler. Thank you so much. Dr. Heinrich?
Okay, can you see my screen now?
Yes.
Okay. Let me know if it doesn't advance. It's a pleasure to be with you today. Ironically, for the same reason that we can't meet together is the reason that I can appear virtually, because today was supposed to be my nephew's wedding reception that I was going to attend instead of LifeFest, but that was canceled. He still got married last weekend in his backyard, but hardly anybody was there, and we hope someday to have a more proper celebration. So I'd like to update you on 3 projects that we're doing, just to kind of give you a flavor of the type of work that we're doing. So the first project is by one of my graduate students, who's second-year graduate student, Homa. And this project has to do with what we call the quiescence or persistent phenotype. So this is the results of a clinical study that was done in France that you may be familiar with. And this went back in the early 2000s when imatinib was first being used. And it asked the question, do you have to keep taking your imatinib if you have metastatic disease? And so this was a randomized study. And so this is the 3-year outcome data, or maybe it's the 5-year. It's hard to, I can't remember because they look the same. But basically the question was, do you have to take imatinib if your tumors responded for 5 years? Why can't you stop? Because your disease is controlled. So this is eliminating everybody whose disease was not controlled for 5 years. So these are people at 5 years that are doing well. And they're randomized to either keep taking drug forever, that's a continuation group, or there was the stop or interruption group in which patients stopped taking imatinib, but had to restart it if their tumor started growing. And so this is looking at kind of the failure rate. So at the time of the randomization, everybody's disease was still being controlled. And what you can see in the red curve is that people who continued to take drug for their metastatic disease did much better than those patients who were randomized to stop, as almost half of them within 4 to 6 months had their tumor started regrowing and they had to restart drug. And basically everyone by a year and a half or 2 years had to restart. But these curves are actually showing 2 different things. So if you keep taking your drug for long periods of time, and then your tumors start growing, that's resistance. So your tumor has figured out a way to overcome the drug. So we talk a lot about resistance, and that's why we keep developing new drugs. But this curve is telling us something different. This is what we call persistent. So a tumor cell that has been treated for 5 years and is controlled can have some type of hibernation strategy in which it can live basically indefinitely, as long as you don't stop taking the drug. And so the question is, and this is what keeps us from curing GIST. So if we could treat it for 5 years and you could stop and it never came back, we would call that a cure. But because you can't stop, we can't cure GIST in the current time. We can only aspire to control it indefinitely. So the question is, what drives persistence? And the short answer is we don't really know. But we can model this in the laboratory. So this is just T1 cells. So this is, Jason just spent a lot of time talking about this. It's a very widely used cell line model. It's a KIT exon 11 mutant GIST model. And so what we're looking at is cells, and this is the cell number in a culture. So basically over time, we're counting how many cells are still there. And if we treat the flask with imatinib, then after 7 days, we get down to a pretty small number of cells left. Although this was a big number, so we're still down to thousands of cells. And then if you stop therapy, you see the same thing that you see in humans. That is, if you stop the treatment, the cells will start growing, and after another week, you're back to where you started. But if you continue the therapy, then you can control this number of cells. So this is exactly what we see in humans. For convenience, this was a 7-day assay, but we've done longer assays. People have even done things where they've grown these kinds of cells for 6 months or a year in imatinib, and if you take away the imatinib, they start to grow. So we have a model in which we could try to figure out what's going on here. And so Homa is a 2nd-year graduate student, so right now she's been studying for qualifying exams, which is sort of this grueling torture thing that PhD students have to go, where they have to make up a project and defend it. And it takes months and months, and at the end, then they can finally go actually work on something meaningful like this. So our idea is to try to identify a therapy that we could apply to cells after they've been made dormant by imatinib. What could we give them that would kill them off? And so, the simplest idea would be to do a drug library, and we've done kind of pilot experiments with Jason down at this big screening lab. It's a really cool place down in La Jolla. But there's other ways we can get to this idea. But the goal is that if we could get rid of the quiescent cells, this could be a cure. And so, stay tuned as Homa proceeds through her graduate studies. The second project is looking at D842V mutant GIST. So as Jason showed, and as David Josephi probably went over yesterday, if you attended the GIST 101, KIT mutations, KIT mutant GIST is the biggest piece of the GIST pie, but other pieces of pie that are important are the PDGFRA mutant slice and the SDH deficient slice. And so PDGFRA D842V has long been a problem because that particular form of GIST has been resistant to all of the drugs. So historically, it's been resistant to Gleevec and Sutent and Stivarga, also known as imatinib, sunitinib, and regorafenib. And so we have these cell lines that Jason and other investigators have used, which are KIT mutant cell lines. But up until recently, we haven't had a PDGFRA mutant cell line, and nor have we had much in the way of SDH deficient cell lines as well. And so we wanted to make a model of the D842V mutant cell line, but we wanted it to be a GIST cell line. And different people have tried different ideas, taking tumors out of humans and trying to grow them in a flask, or try to grow them in mice, but that's been pretty slow. After 20 years of trying, we still have 0 of those cell lines for the most part. And so what we decided to do is start with an existing GIST cell line that has a KIT exon 11 mutation, and put in D842V and then subtract out KIT exon 11. So we know that this is a GIST cell line. We know, so that represents the biology we care about. And D842V turns out can drive the cells just as well as KIT exon 11. But we have to sort of sequentially put one thing out and then take the other thing out. And so we developed a cell line. Actually, we've developed like 6 of them, but this is was the first one that has D842V. And so the question is, does this cell line represent what would happen in humans? And so the big question would be, is it resistant to imatinib, but sensitive to avapritinib, also known as Avakit, which is a new FDA approved drug, which is very potent against D842V? And the answer is yes, our cell line now models that. So it's relatively resistant to imatinib, and it's very sensitive to avapritinib, which used to be called Blue 285. Again, this is showing 100% alive. This would be 0% alive. And so we mostly kill things off using avapritinib, but we're unable to kill it off using imatinib. So then we can now go into the clinic in patients treated with avapritinib. If they ever develop a resistant tumor, we can biopsy it, or surgeons can remove it, or we can do circulating tumor DNA. And if we find new mutations, we can now model them in our cell line and try to figure out, well, if it's resistant to avapritinib, what other drugs might it be sensitive to, with the possibilities, at least in the literature, being ripretinib or crenolinib. So this is kind of a model of some of the work we've done, showing that these mutations in PDGFRA are sensitive to avapritinib, but we can find mutations in patients that are secondary mutations that make avapritinib not work. Unfortunately, they mostly make other things not work as well. And so we're still trying to figure out how to get around that problem. Of course, we have our 5,000 or more drug library, so we can take a look at that. And we've done some of that work, again, with Dr. Siklick's help, and we were able to narrow down 5,000 drugs into 68 drug candidates. Interestingly, many of them were the type of inhibitors, the PI3K inhibitors, that seemed exciting for the cancer-associated fibroblasts, so a little bit of synergy between the projects. And my final topic is to talk about, it's more of a diagnosis and genetic counseling problem that we see in SDH, that a different graduate student, the last work was done by Lily Klug. This work is being done by Jason Kent. And so SDH accounts for about 10% of GIST. SDH stands for succinate dehydrogenase, which is kind of hard to say, which is why we say SDH. And it's an enzyme complex, so it's totally different than KIT and PDGFRA. It's really an energy supply and metabolism center for the cell. And it's made up of 4 parts, which are 4 different proteins that come together. So we have SDHA, SDHB, SDHC, SDHD. So it's sort of like a Lego thing of 4 things that have been combined together. It's located in the mitochondria. It's important for normal metabolism. And enzymatically, what it does is it converts something called succinate to another something called fumarate. Now, you could think about this really, that this complex is like an engine or a machine. And so the 4 subunits have to work together in order to convert succinate to fumarate. But if something happens and mutates and inactivates part of the complex, then our nice machine doesn't work because we're missing a gear. And the consequence of that is we can't convert succinate to fumarate. And then the amount of succinate in the cell grows, increases, and that actually ends up poisoning the cell in ways that are too complicated to explain. But it's this accumulation of succinate that actually leads to cancer. Now, in order to kind of understand what we're doing, I need to maybe repeat something that might be very simple for those who know biology. But in order to understand what we're talking about. So the central, basically is called the central dogma in biology, is that DNA, which is your genome, it's the genetic information, is a code so that you can make protein. And then depending on how the protein works determines the function, whether it works correctly or not. So for example, here is a sequence of DNA, what we'll call the wild type, which means it's normal. And let's say that we could translate this into a language, and the language means the cat is fit. So we have our happy little cat here. But let's say we change one letter and the DNA, and it changes the protein and it changes the function. So now we have the cat is fat, not the cat is fit. It's almost the same thing except for one letter, but in English, of course, it has a completely different meaning. And then there's also other mutations in which right here, this A highlighted in red is different. And the sentence reads a little bit different because now it's a capital C instead of a small c, but really, we are able to understand the meaning, the cat is fit, so we still have a happy cat. So what we care about is the idea that if you have an SDH mutation that you inherited, that can be bad because it can lead to a very high risk of developing cancer over your lifetime. So this is looking at patients who have a SDHB mutation, that's the harmful kind, and looking at the percentage of patients who will develop cancer out to age 60 or 70. And as you can see, the number gets quite high, that about 80% of patients will get an SDH deficient tumor by age 70. Which includes things like GIST, which is why we're talking about, but there's also other cancers, cancers of the kidney, cancers of the adrenal gland, or weird cancers of nerve tissue called paragangliomas. And we see the same risk here in the SDHD population. And you can, if this person, sort of the father of the family, has the mutation that is associated with this risk, then there's a 50% chance that each of his children inherit this mutation and have the same risk. So, and what happens is in healthy people, well, when you're born with one mutation that's harmful and one that's normal, you're in this situation where one of the machines is broken. So this is a copy from the father. Oops. But the copy from the mother is normal, the machine works, and you actually only need half the machinery to work in order to be healthy. But over time, cells can make a mistake and they can lose the mother's copy, and you end up with the defective machinery, and then that leads to cancer. So in families like this, where we know there's a harmful mutation identified in this person, we'd want to screen his children, but also his brothers and sisters and his parents and his grandparents. And if it goes to his brother, it can go to his nephew. So it can turn into a large number of people who are at risk. On the other hand, these people shown in blue have no increased risk. And so we don't want to scare them or do a bunch of medical things to them because they have no increased risk. So the question is, which mutations result in loss of SDH function? And the answer is mostly we don't know. So of all the possible mutations in the SDHA or SDHB, the ones that are known to cause cancer are shown in red. The ones that are shown in green are known to be benign, to not cause cancer. And the ones that are shown in gray are what we call variants of unknown significance, which means we don't know if they're good or bad. And so we can't make any recommendation. We're not going to screen your family because we don't know what we do with the results. And we're not going to do any cancer screening on you beyond normal procedures because we don't know if you're at an increased risk. And so what we're trying to figure out is how could we reduce the size of this variance of unknown significance. Now, to put this in the proper perspective, 1.5% of the US population has an SDHA or SDHB variant, the vast majority being variants of unknown significance. So that means that somewhere around 5 million people, if they had their genome sequenced by 23andMe or Ancestry.com or for medical purposes, would be identified as having an SDHA or SDHB variant, but we wouldn't know what to tell people. And so the VUS problem is really, what if we don't know the language? So I showed you before, DNA goes to protein and protein goes to function. But the problem is we don't understand the protein language. So in these two sentences here, they look kind of almost the same, except for at the end, but I don't speak Romanian, so I don't actually know what these mean. But it turns out that this means the cat is fit, and this means the cat is fat, at least according to Google Translate. And so the VUS problem is that we know what the mutations are, but we don't know what they mean, and so we can't go forward. And so what Jason is working on is instead of trying to guess one at a time, we actually, there's ways, and I won't go into all the complicated ways, that we can model every possible SDH family member. So there's 13,280 possible SDHA mutations. We can model all of them. And likewise, there's 5,600 SDHB mutations because it's smaller, 3,380 SDHC, and 3,180 SDHD mutations. So in all, there's slightly more than 25,000 mutations. And so his project as a graduate student, at least for A and B, is to model every particular mutation and to figure out if it affects the function of the protein or not. And so right now, as we sequence people's genomes, we mostly have missing information. We don't know what the gray boxes mean. But if we were able to convert this sort of to a battleship type matrix in which each box represents a different possible mutation at a different possible location, we could, by doing all this profiling, create an encyclopedia. And so if we knew that you had an SDH mutation or variant in your normal DNA, we could look it up. So in this case, we look it up and turns out red means that this could cause cancer. And this means that we should provide you genetic counseling. We need to test other family members, such as your children, your siblings, your parents, your grandparents, aunts and uncles, nephews and nieces. So sometimes this turns into going from one person goes into immediately into dozens of people who are potentially at risk. And if we identify your risk, there's things that we can do lifelong that would detect a GIST in its early form, so that, you know, our wonderful surgeons like Dr. Sicklick could go in and do a very small operation, take out a little part of your stomach, remove a 2-centimeter tumor and be done with it, rather than wait till it's a 5 or 10-centimeter tumor and it's metastasized. So knowing which ones are the bad mutations allows us to decide who needs all of this extra stuff. But likewise, most of these variants will turn out to be benign, and so we don't need to do all this stuff. So if we find from 23andMe or however we sequenced your genome that you have an SDH mutation, but we look it up and it's benign, then we know that it has normal function. You have no increased risk of cancer. You don't need to do any cancer screening, and you don't need to have any family testing done. And so that's sort of the goal. And so this is just a list of people over the last 20 years that have worked in my laboratory on these projects. And these are some of their faces. Thank you.
Thank you, Dr. Heinrich. Thank you so much. So we are in the Q&A portion, and I have several questions here. I'll start with you, Dr. Sicklick. What % of rectal GISTs have a different surgery as a result of neoadjuvant Gleevec?
That's a good question. I don't think we know the exact answer to that question, because it really depends upon the size, the location, where it is relative to the sphincter, et cetera. That all those factors together, play into that. And so, it's potential that even if you shrunk a tumor down, if it's very close to the sphincter, that still may play a role in having the same operation, but perhaps might be less likely to, or more likely, I should say, to get the entire tumor out. So it's a difficult answer to question to answer sort of just globally, but you know, on a specifically, it's on a case-by-case basis.
Thank you. Dr. Heinrich, in your experience, could you say the longer a GIST patient goes with NET scans, the more likely this trend will continue?
Well, you know, it kind of depends what you mean by that. So if you look at the curves of people like an imatinib for metastatic disease, you know, about half the people will have a failure of imatinib by 2 to 3 years. So that we're down to 50% of people. But to get down, the next 50% takes like 7 years. So it takes out to 10 years to get down to 25% or so. Well, maybe a little bit shorter than that. But so yes, after a certain point, you know, your odds are better that your next scan will be fine. But, you know, it doesn't guarantee that, you know, inevitably something won't be wrong in the future. So I think so put it a different way. If you start imatinib for metastatic disease, your biggest risk of failure of the drug failing you, is in the first 2 years. The next biggest risk period is the next 8 years. So making it to 2 years is kind of a big deal. Making it to 5 years is a big deal. If you make it to 10 years, you know, you're kind of in the 10% of people or something like that. So I guess that's how I would look at it. I mean, we do have people, you know, just to be positive about things, we do have people that we started back in 2000 who are still on the drug and have disease control. So that's a, you know, minority of the people that started our original study back in 2000. But still, every day they're setting a new world record for how long you can control your GIST with imatinib.
Great. Dr. Siklik, are there many GIST patients taking 100 milligrams and 200 milligrams? How effective are the small dosages?
That I'd probably defer to Dr. Heinrich in terms of, I'm assuming they're referring to imatinib.
Yeah, it wasn't clear. So yes, I would
Yeah.
assume imatinib. Yeah.
You know, in trials, we've never really gone below 200 milligrams. I think in studies, if we had to go below 200 milligrams, or sometimes even below 300 milligrams, you would have been off the study. In my practice, I've seen very few people that can be treated with doses that low. Generally, they either have something wrong, like severe kidney disease, or they must just have very different metabolism of the drug, because we can look at their drug levels and see that very small doses still keep them therapeutic. But the vast majority of people, if you drop below 300 milligrams, I would be very worried that it won't work for you.
Staying with you, Dr. Heinrich, my doctor has asked me to stop taking imatinib after 2.5 years due to severe side effects. If GIST comes back, would imatinib still be effective?
So this is in the adjuvant setting. So someone's had surgery. I'm assuming they had it removed. They're taking it. They were trying to get to 3 years. Didn't get quite there. What we know about that for people who go off adjuvant therapy is that if your tumor comes back more than 6 months after you stopped, then it's sort of the persistence thing I showed you, which that the tumor can survive the imatinib in some cases, but it's still sensitive. So we can restart the medication. So yes, it would likely work, but unfortunately, it also likely would produce the same side effects. And so, you know, it'd be a big discussion with your oncologist whether or not you think you could then take the drug again indefinitely, or whether, you know, is there something different we could do for your side effects, or, you know, we don't really like to switch to sunitinib, and, you know, when imatinib still could work, but sometimes for side effects, we have to.
Dr. Siklik, do you have any recommendations for a specific diet for a GIST patient?
I do not have a specific diet. I think it's really just a healthy, well-rounded diet, you know, avoiding, you know, large amounts of fat and sugars, and, you know, but not any one specific that is tailored just for GIST.
Do you have any good suggestions for minimizing side effects from taking the medication, like for shoulder pain and back pain in the back of the head? Is it common for just patients to have those types of pain? And I'll open that up to either one of you to respond.
Well, those would be kind of unusual for imatinib side effects, but they're kind of common in primary care practices. If you just go in, like see what's common, people have headaches, people have shoulder pain. I've had my shoulders injected with steroids for tendonitis. I've had back surgery. So, those kinds of things are common. So I don't know that they're due to imatinib per se, and perhaps investigating other possibilities. I mean, they may be, but there's lots of common things that cause those.
Dr. Heinrich, or Dr. Sicklick first, I'm sorry. What is the criteria for deciding to do mutational testing?
That's a great question. I think that there's several criteria. One is that the patient is going to be considered for treatment with one of these TKIs in the setting of imatinib versus now with the FDA approval of avapritinib. Clearly, there's differential sensitivities to these drugs, and so to give a patient with a PDGFR alpha D842V mutation imatinib at this point, I think is the only potential there is toxicity or side effects without any potential benefit. So I think it's going to become increasingly important. We've got a publication in Press right now that will be coming out at some point in the next several months, basically showing that looking in the metastatic setting, asking the question, is it cost effective to do mutational testing in patients with metastatic GIST as a first step in terms of determining their drug therapy? And essentially the short answer is yes, it is actually more cost effective just from a pure cost standpoint, not even relating to a drug that's actually working as opposed to being on a drug that's not effective, is that it's actually more cost effective to sequence patients so that you tailor their therapy appropriately for metastatic GIST.
Thank you.
So I would agree. So any GIST that you're thinking about that you would give the patient a drug, you should know mutation testing. I think the only thing I would add on is if you have a gastric GIST and there's any suspicion that it might be SDH deficient, then we should do mutation testing in that case, because 80% of SDH deficient tumors are inherited. Which means that you could pass it on, and sort of the thing I showed you, family trees. So we even if it was a small 2-centimeter tumor and we know that it's not coming back, we want to identify that you're at risk for something else coming back, or that other family members are at risk. So we would want to investigate that a little bit more, even if the surgery went well and there was no need for therapy.
Thank you.
Just one other thing to add is on Monday at the SCH session, I'll be talking about, we've sort of come up with an algorithm potentially to predict which patients are at higher likelihood for gastric GIST of having an SDH versus a PDGF versus a KIT mutation. So that data potentially will have the ability to help inform who should have mutation testing to rule out PDGF versus an SDH mutation as well.
Great. Dr. Heinrich, patients attribute every change to their bodies to Gleevec and imatinib. What are the confirmed side effects, and how can we tell whether a particular change we are experiencing is directly related to the medication or not?
Well, that's a complicated question, because if I gave you a consent form for imatinib, it would have 5 pages of side effects, and but a lot of them are probably not actually related to imatinib. It's just that if you give a drug to 1,000,000 people and some of them get kidney stones, all of a sudden kidney stones shows up as a side effect, even though if you took 1,000,000 people, some of them would have had kidney stones already. I think that things that could show you that is, if you get confused about things like that, I mean, generally sometimes if I can't figure it out, I'll just say, just stop the drug for 2 weeks and most things will be gone in 2 weeks. And if things change, then yes, that's probably due to the drug. If things don't change, like I still have diarrhea every day, well, maybe that's because you had a Whipple operation and you're not taking your Creon, or because part of your small bowel is resected or whatever. So, sometimes it's pretty tough to discern those types of things. So, stopping imatinib is reasonable to figure it out. I mean, if you fell and broke your hip, we would have you off the drug for a couple of weeks. So I think sorting out side effects, we can safely stop the drug for a few weeks if we need to. It's kind of hard to say, like, let's say you've been on the drug for 10 years, and my sad experience of being 10 years older than I used to be is that my body is different than it was 10 years ago. And, I'm wearing glasses now and I didn't used to wear glasses. So things can change over long periods of time. And, it's not always due to the drug. And so I think that, working with your doctor, trying to understand the side effects and, trying different strategies, sometimes, although there's not any diet that's better as far as outcome, I think some diets work better for people who've had parts of their stomach or small bowel removed, or they're on imatinib and, they can't eat the way they used to, but we can find things, food that they can still enjoy that doesn't cause as many GI side effects. So I think it's a big conversation to have with your doctor.
Great. Dr. Siklik, if the calf drug gedatolisib, sorry for my pronunciation, does work, would it be given with imatinib in the adjuvant setting after primary tumor was removed?
That's a good question. I think first things first, obviously we've shown this in the cell lines, we've shown this in the mice, but the next step would be looking at this in humans. It's not FDA approved drug at this point. I think the next step would be to investigating this first, most likely in the metastatic setting, generally where we would think about doing this first, because we can have disease that we could potentially follow and see if the drug is working in combination with imatinib. The adjuvant setting is a little bit different in that you're taking a drug where it works for this disease and then applying it to patients where they've had their disease removed, and you're expecting that this is going to decrease the risk of it coming back. And so it's a different question that you're asking, but because you're asking whether this is going to prevent disease recurrence as opposed to can this actually treat the disease itself. So generally, we would think about doing a drug trial in the adjuvant or postoperative setting once we know it's effective in the most often the metastatic setting, or in the preoperative setting where we could show that it was shrunk the tumor itself, where we actually had something that we could measure and say, this drug is working.
Thank you. Dr. Heinrich, is pasopanib recommended after resistance to imatinib, sunitinib, or regorafenib? And if yes, then how to deal with pasopanib-related fatigue?
So in the US, ripretinib would now be approved for the 4th line, and your insurance would pay for that. Or if you have trouble with the copay, the company would help you with that. So I generally wouldn't use a drug like Votrient or pazopanib, which is off label until we sort of exhausted the existing therapies. Fatigue with all these drugs is a problem. Sometimes we can lower the dose, although we have to be careful about that, as we've discussed. In my experience and in lots of other cancers, it's been shown that being active actually helps you be less tired. It seems kind of paradoxical, but having some type of exercise program, even if it's walking, and even if you can only walk a quarter mile, maybe do that and then try to work up to a half mile. And so you know, your body's designed to move, so moving makes you feel better. It also makes you physically tired, which is different than mentally tired. So it helps with your sleep, which kind of helps you feel better the next day. So, you know, I encourage people to be as active as they can within the limitations of their health and try to, you know, become more active than they currently are. And I think that's generally the best strategy. But it's all it's all relative. When to what you want to do. It makes me laugh every time I think about this. But one of my patients said, this imatinib is making me really tired. So, okay, well, what do you do? Well, I'm an attorney. I work 80 hours a week and I run 6 miles a day. And it's like, okay, that sounds kind of tiring. So she actually ended up reducing to 60 hours a week and felt better. And so really it had nothing to do with the drug as much maybe as it had to do with lifestyle. So she was maybe a little bit on the too active side of things, but trying to be active, I think, is the best strategy. If the drug is just making you so fatigued you can't function, then we're going to have to lower the dose or find a different drug.
Great. Should people with exon 11 have family members' DNA checked? No, I'll open that one up.
In general, no, unless the family history says somebody else has a GIST. I mean, I think in my 20-year career of sequencing thousands and thousands of people in GIST, we've identified like 3 families. And mostly those were already self-identified because multiple people had GIST. So as a routine practice, no. But I did have one case this year where the father had GIST with a KIT exon 11 mutation. And then he identified some unusual things in his daughter, which led us to suspect that maybe he did actually have this situation. So talking about your family history to your oncologist could kind of identify the extremely rare, I think there's like 50 known families in the world that need that kind of testing.
Okay. Can you discuss generic imatinib versus brand Gleevec?
Is that me again?
Yes.
So in general, we were kind of more fearful of that transition over, like 4 or 5 years ago. In my experience of hundreds of people having to be switched because their insurance company said so, that I would say that 95 people out of 100, they can't tell a difference, and I can't tell a difference. And I don't think I've ever seen a case where I thought it worked less well, just sometimes the side effects were different. One of the annoying things, the way our healthcare system works is that your insurance will say you should get a generic because it's cheaper to them, but your copay is $2,000 and they're not going to help you with it. And so we have to do what I call the Gleevec shuffle, where write for imatinib, get a $2,000 copay, switch to prescription to Gleevec, get it denied, then write a letter of appeal so that it can be denied twice, so that you can then, if you don't have too much money, go to the patient assistance and get Gleevec, at like no or little cost. So it's, sometimes the generics, even though they're cheaper, are more expensive to the patient, which I mean, I don't care what the insurance profit margin is. I only care about what my patient, that they can afford the drug. So it is complicated sometimes financially. We have a whole department of people who's called the medication assistance program, whose job is to fill out forms all day long to help people get medication assistance. But medically, I don't discern that there's much difference between them. But it is confusing since there's, some countries like Canada or Europe, I think there's, 5 or 6 generics. So it would be, I think, disconcerting to have a different pill shape and bottle every single month if people switch that often.
Okay, thank you. Dr. Cyclic, my GIST was discovered 7 months ago with 10 centimeter size, tried imatinib 400 milligram. After 3 to 6 months, it has gone to the liver. Is that common?
Diana, can you repeat that again? I'm freezing right now.
Okay.
I can't hear anything.
Let me see if I can pull it up.
I can hear you now. Okay.
Okay, perfect.
All
I'm just trying.
I heard was 10 centimeters.
Started on imatinib after 8 months or so. Now there's a liver metastasis. Is that common?
Exactly. That's the question. Thank you.
Sorry, that cut out again. I started on
Okay.
imatinib and then
Do you want me to answer?
Go ahead.
Go ahead.
Jason, this is someone who was getting neoadjuvant imatinib, and then after, before having surgery, a liver metastasis was noticed after 10 months of therapy. So it's
Okay.
not very common. So there's a couple possibilities. One is that this is a type of tumor that's not sensitive to imatinib, so like an SDH or PDGFRA D842V. And so if we didn't know the mutation going in and it's a resistant type, then yes, that would be a common outcome potentially. The other scenario is that sometimes we don't appreciate liver metastasis because they blend into the liver until we give imatinib and we kill them. And then all of a sudden they're very easy to see. So sometimes we see people and we don't think they have liver mets, or we think they have 3, and then we give them a drug and all of a sudden we realize, oh, well, there's 20 because we killed them all, or at least they look dead. So I'll kick it over to Jason at this point.
Okay. Thank you.
I can thank you. I heard most of that. It keeps cutting in and out for me. I apologize. Yeah, I agree with what Mike just said in terms of sometimes the response actually brings out the disease you can't see. Certainly it is, the case sometimes where we do see that, but it's not very often that we would see somebody that we would know has a imatinib sensitive mutation that then suddenly develops new mutations that, in that time frame, but it is possible. In general, in terms of like, when do we think about resecting metastatic disease? I mean, in an ideal scenario, we would want to resect metastatic disease in the setting of patients that are receiving therapy and their therapy is working, or they're responding to treatment. That would be our sort of a preferred, option. Second best option would be is if somebody had majority of their disease responding, and it just happens that there's one mutation, or there's one tumor that's not responding and or growing, that would be another situation where we would consider doing a resection. The sort of the least favorable scenario would be is somebody that has disease that is growing in spite of being on treatment. And that's obviously suggests that the drug's not working, that they've developed a resistance mechanism, or they've had primary resistance, meaning that they had a PDGF mutation, but yet they were receiving imatinib, and therefore imatinib's not going to work. So it kind of goes back to the earlier question that somebody asked about genetic testing, and goes to show you the importance of doing that before you start on a drug, especially in this era where we can start really tailoring the therapy and personalizing it to that individual patient, rather than everybody gets imatinib because a majority of just have a KIT exon 11 mutation, for instance.
And the flip side of that in this setting of what we're trying to shrink the tumor is, if we think that we can't shrink the tumor, maybe we should just do surgery first rather than, try things that are unlikely to work.
Agree. In terms of when to think about operating, can you hear me?
Yes.
Yes.
And no.
No. You've frozen. Okay. I think we have a time for one more question. It looks like Dr. Sicklick is frozen. Dr. Heinrich, do we know of any just patients that have had the COVID-19 virus?
I don't personally know of any of my patients. I know some of my CML patients have had it, but I don't know of any GIST patients. But, Oregon's kind of low prevalence, so that doesn't really mean so much. I think we're up to like 10,000 cases, and that's some states one day. That's all of our cases thus far. So I think we just have lesser amount thus far. The question comes up a lot whether or not, whether like the therapy going to make COVID be worse for you. And we don't think necessarily the drug will affect your immunity. We don't think it makes you more susceptible. And we don't necessarily think it would make things be worse if you did get COVID. I think what's the bigger determinant is what is your underlying health. So, if you have diabetes, if you have heart disease, if you have lung disease, if you're very elderly, then those are big risk factors for COVID being more severe, kind of irrespective. Or, if your cancer is very advanced and you're doing poorly cancer-wise, losing weight, and then it probably would be much worse for you.
Dr. Siklik, would you like to chime in on that question? I know you just joined us back.
I joined you back. Apologies. I don't know why I got kicked out.
Okay.
Maybe you didn't like my answers. I just got the tail end of what Dr. Heinrich was saying in terms of the answer. So I don't know what the question was, but I'm assuming it was something of relation to imatinib putting you at risk for COVID, something like that, I'm assuming.
Yeah, the relationship of COVID with just patients.
Right. I think the key is, we were talking about before the session started, is being vigilant, wearing a mask, being careful about exposures. I think right now in a lot of places, there's a tendency to sort of let your guard down, if you will, and people are congregating with sort of the assumption that this is all behind us, and it's clearly not. If you look at the rates of, in many locations around the United States, that the rates are starting to go back up again, as is the mortality rates. We're seeing here in San Diego, I just read this morning that the ICU beds are starting to fill back up again when they were starting to empty out. And so I think we need to remain vigilant about this. And as individuals that have just it in and of itself, to our knowledge, doesn't put you at risk for it anymore, but certainly other risk factors, as Dr. Heinrich mentioned, certainly can if individuals do have those. And knowing statistically that just tends to occur in patients that are in 60s and older, that they're just based on age and other comorbidities, the risk is higher. So stay safe, stay vigilant is what I would say.
And I saw in the chat that someone said that they had and they're on adjuvant imatinib and they were diagnosed with COVID, and their oncologist said hold their imatinib until after their quarantine. I think that's quite reasonable. I would do the same. I don't think it puts you at increased risk, but for adjuvant therapy, being off it for a week or two is completely, I think, safe given the situation. So I would agree with that. And I guess there's at least one GIST patient who had COVID, so now we know.
Thank you so much, Dr. Heinrich and Dr. Siklik, for your time today in your presentations and answering the questions. We are out of time for this panel discussion. Laura, did you want to chime in?
No, thank you, both Dr. Heinrich and Dr. Siklik. You're always there for us, and we truly appreciate all you do for our GIST community.
Yes,
Thank you
absolutely.
very much. Our pleasure.
Yes. So if you're joining us for the Oncology Surgical GIST Expert Panel, please go to the LifeVest agenda email that you received and click on the link for that panel. It is a different Zoom link than the one you used for this research panel. Or if you are viewing us live via Facebook or our YouTube channel, just go back in at 2 p.m. Eastern Daylight Savings Time. So for those of you that did ask questions during this panel that we didn't have time to answer, come back at 2 o'clock and we'll see you then. Thank you, everyone.